Okay, I think I started. Okay, so welcome back again for this. It's going to be most likely another exciting session on on protein design, right? Protein engineering and protein design. So in the last lecture, we talked about the fundamentals and the basics of proteins. You know how proteins are made and what are the structural elements, what are the fundamentals, why proteins form shapes and size and why function, and what is the relationship between the structure and the function. So mostly we talked about that. And so today we'll move, and then obviously we also, give a little demonstration on how to use PyMol and things like that. And so today we'll moving slightly away from um, we'll moving away from the fundamentals, and we will talk about I'm still trying to struggle, let me see, sort of let me I'm trying to keep this okay, anyway, that's fine. So today we'll talk about the engineering side of like making new proteins and how we can engineer proteins for therapeutic and for medicine basically. So in last class I forgot to mention about a book on protein structure, and so people who are really interested understanding protein structure, what they are built of, and what kind of different kind of protein structures available in the functional part. This is one book that you might like to follow in longer run. I thought I'll just mention about that. This is a wonderful book. Yeah. And I have to. People can people mute themselves? I have to stop sharing and then mute all. Mute. Okay. Yeah, so I keep telling people that please mute yourself, otherwise it creates a little bit of disturbance here. Please do that. Let me share again. So okay, so then and so today we'll be mostly talking about we'll walk through some of the protein structures and see how proteins look like. How obviously we talked about this. Nature has like like full of like millions of proteins, and then it is impossible to talk about all of those obviously, and they all have different functions and different shapes and things like that. And we'll just walk through a couple of proteins, a couple of different machineries before we start talking about making proteins, engineering proteins. So one of the things that in the last lecture I briefly mentioned, one of the things that is very abundant and so important in protein structure is the loops, right? And obviously the beta strands and the alpha helices, they support, they give the foundational the structure, so that the protein can form a three-dimensional shape. But many times it is a loop which is very dynamic, because the loops do not have a rigid structure, and most of the functions actually come from the loops. And here this is an example of antibody, where we'll come to it, what the antibodies are, and where this antibody has all these loops, and the loops are basically connecting all these secondary structures, beta strands mostly here, and there are alpha helices as well. And so you might think that maybe the loop is only good for, like, for example, here this is a loop, and it's only good for connecting these beta strands and the alpha helices, right? And that is true, it's a connector, but also most of the function actually arise from there. And the reason why the function comes from here, because they are very floppy, and they are basically very easily mobile, and they can move around, and they can change their shape and things like that. And because of that, this class of protein called antibodies, which are I'm pretty sure most of you know, Antibodies are produced by our immune cells, beta cells, sorry, not beta cells, B cells, and then obviously B cells mature to plasma cells, and so they produce this antibody to fight diseases in our body, right? And that's part of our adaptive immune system. And because the antibodies have antibodies are the primary warrior, right? In one sense, they are the main fighter of disease in our body. And people, ever since antibodies were discovered, scientists have been trying to repurpose these antibodies so that it can fight disease better. And in the context of medicine, I keep telling that like if we have to build our immune system, if we have to re-engineer our immune systems, and most likely one of the main main player that we want to re-engineer is the antibodies, because they are at the forefront of fighting disease, right? I mean, that when you have COVID, when you got vaccinated, and what vaccine does is that basically it'll make our B cells make more and more antibodies, which can basically neutralize the viruses, right? And so because of that, there has been huge interest in engineering antibodies, and then I cannot it's impossible to tell the importance and how much is going on around the world for antibody engineering, and this is a huge discipline that has emerged in the last 30 years, and my guess is that it will stay for next, I don't know, many decades most likely. In slight more details, the antibodies have this protein structure where you have this called constant region, and these regions basically don't change, and then there is this variable region. It's called V, and this variable region where here, and there are two chains, you can see the blue chain and the slightly yellowish chain, right? And here in this region, the tip region is so variable that you can re-engineer antibody, and that's what our body does. Whenever there's a new germ, new something coming in, our body need to re-engineer the antibody so that they can fight against this new invasive whatever enemies, right? And so that engineering happened the way there's a mechanism called somatic hypermutation and all. I'm not going to go into details of immunology here, but that re-engineering in our body, the re-engineering happens mostly at this region, because these are heavily as you see, there's full of loops, and all these loops, our immune system try to mutate and re-engineer so that they can fight with anything that comes in, right? And so the same principle is used when scientists try to re-engineer these antibodies, and try to basically engineer these loops and mutate or sort of evolve them so that they can bind to all these invasive enemies and things like that. And so there's so many different ways that antibodies can fight disease. Is that, for example, if there's antigen is a molecule, a protein, for example, secreted by some kind of germs, and that can be antibodies can neutralize those antibody. Yeah, somebody also, I'm sure that you're not intentionally trying to draw here, but I think you have to be careful with your setup, because there's this drawing coming here. So the antibodies can neutralize those antigens, and then there is also different ways of basically these antibodies can also bridge a cell which is harmful for our body, and the antibody can bridge to a lot of immune cells which can kill these cells. And this is one of the major ways now these days that people have been trying to kill cancer cells. Imagine this is a cancer cell, and then there's T cell or some B cells, and if you can engineer antibody which can bring these two together, you can also kill the antibody cells, because these cells are sort of different kind of warrior, right? Different kind of fighter. And That's one use. And then the other way also, like, the antibodies can also bring other proteins. For example, the complement system, it can engage the complement system into a cell, and then again that it can kill the cells. Again, this is like extremely brief. I'm like, this is like so brief. And as I said, antibodies are one of the major medicinal candidate, and there has been huge effort for engineering antibodies. And I would say, like, the entire pharma industry, entire pharmaceutical companies, and all this biotechnology industry, I would say, like, majority of the projects right now, majority of the efforts are towards engineering antibodies. And within last 20 years, 20, 30 years, there have been at least hundreds. Right now, obviously, the number might be way higher, but at least 100 antibodies in therapeutic use. And again, these are not only for one disease, starting from cancer to autoimmune, and there's so many different diseases that you can actually re-engineer antibodies so that you can fight disease. For those who are really interested in diving deep into antibody engineering, it's not, as long as you understand the fundamentals, as long as you know the basics, the protein structure, and how they look like, where the function coming from, how you can re-engineer them, mature them, and those kind of stuff. This is incredibly interesting topic, and at the same time, it has profound impact in humanity. So I would suggest those who are really interested in going in a path where you want to be an antibody engineer, for example, I would strongly suggest that you read some literature, find out more work, and if you just Google like review on antibody engineering, you'll find tons of those. There are books written on antibody engineering. So and then the tools, last class I showed how to use Pymol, and all you have to do is, like, download Pymol and get some antibody structures from the PDB data bank, and you can play around, and you can try to, like, mutate by yourself. I mean, obviously you won't be able to do any experiment, but just to familiarize, just to get more concrete knowledge on antibody engineering. There are tons of resources available. So I think it's only about your will and whether you want to learn something before you move on to actually doing some cool science. So anyway, so you can do that. There are lots of resources, as I said, and because we are talking about so many other things, so we are not going to talk more about antibody engineering today. So instead, I would, again, as I said, I want to give a few examples of how these protein machines work, and they're all important from medicinal perspective. This is a protein that evolved in our body, in, I think, not only humans, not only mammals, I think across many, I don't know exactly what, how many species have this protein. And this is a sensory protein. And this protein, what happens, like, when somebody touches you, or even you touch, or even, like, some cool air or any mechanical force coming around you, and you feel it, right? You have a sensation when you touch, you feel you're touching, right? And you can wonder, how is that happening? How is that, like, the feeling of sensation is going to your brain, right? I mean, obviously we take it for granted. The nature built us, and we think we have all the right to exist here, but nature spent millions of billions of years to build us, right? And a lot of this building process actually happen in a very intricate, complicated way, and today we take it for granted. And so this is a protein actually which is involved in this touch sensing, and this protein name is piezo. Piezo is like pressure. I don't know what word it is. I think physics people will know better. Piezo is like, it has to do with pressure, like, and when there is some slight variation in the pressure, and this protein is expressed by all the cells in your body, and when there is some pressure, and what happens is that and then in terms of structure, it just looks like a fan. It resembles with some of the natural things that we have. And so the piezo, what happens is that this protein, it has these blades, and this blade actually rotates when there is a pressure around, and And so if it's just like just like the blades here, like it rotates, and it creates lots of pressure, right? So the same thing happens when there is pressure. These blades will rotate, and during that, what happens is there is a pore here, and when these blades rotate, the pore gets open. And when this pore gets open, there is a flash of basically ions moving in, and that's how there is a difference in voltage. And then obviously that sensory system is full of, like, it's basically voltage thing, right? It's a electricity thing, and because you have this change in your voltage, and so there is a new information going into your brain basically. And the understanding, this protein actually led to the Nobel Prize this year. In fact, Ardem Parapoyan, he got the Nobel Prize along with I think David Julius. And yeah, so I just showed this example how proteins are very intricate. And we in last class we talked so much about protein machines, and these are some of the protein machines, how how simple these machines are, and yet do profound function. And from the medicine perspective, you can think that there are people where people have mutations in these proteins, and when they have mutations, you can imagine all the troubles they may have, right? Again, like we can call that disease or not, that's a different issue. But a person who has mutations in Piazo, they will have lots of problems, like sensory problems, right? And that sensory, I'm not only talking about somebody's trying to touch you and you don't feel it, but also like in your stomach, in your gastro, like the gastric system, there is also pressure sensors, right? During like metabolism, during all this food digestion and all. And so a lot of the time, like pain sensors, right, kind of stuff. And a lot of the time, all of these different like different receptor proteins, they get mutated, and people do not have those sensations. Not only this piezo, I'm talking about other sensory proteins as well. And it is very important to understand these proteins, from structural perspective, what goes wrong, and if there's some mutations, and sort of come up with ways to intervene that, like create new medicines basically. So this is again, as I said, we cannot go into all different classes of protein, but this is another protein. Again, most of you already are familiar. This is a membrane protein, and there are hundreds of membrane proteins, and they're all part of signal transduction in your cells. There's an intracellular side, there's an extracellular side, and extracellular side basically communicate with outside world, right? They will bring messages, bring new information through molecules and ions, and then they will pass on to cells. There will be new gene activities, new expression of genes and all, and that's how it functions, right? That's how living system function. And so this membrane protein, as you can imagine, it has a transmembrane domain where this is like more hydrophobic, right, lipid-like structure. And then the intracellular domain also, it does new function, and same with the extracellular domain, which is obviously hydrophilic, but also it has other molecules so that it can interact with the outside environment. And so that's the function of membrane proteins basically. And obviously there would be conformational chains and for certain triggers, and that's how messages have been sent inside. And then again, for many diseases, this membrane proteins is so important to sort of intervene their functions or even re-engineer some of those proteins. This is a proteasome, and this is again another machinery. In our body, there are tons of different machineries, obviously, and this proteasome complex plays so big of a role in the homeostasis of proteins. So these proteins are made by our cells, right? And then the genes will keep getting transcribed into mRNA, and mRNA into proteins. But what happens when you make tons of proteins, and they need to be degraded, right? And they need to like recycle, otherwise the cell would be clogged and cells would die, right? And so one of the way to declog all these things is to make a program degradation of all the protein that exists within the cell, and that happens in this protein complex called proteasomes, which looks like just like a trash can. Basically, it degrades all the proteins. and obviously there are so many different examples, and this, I think this is the final example I'm giving before we are talking about protein design. And this all this virus. In the last class I mentioned that the level of structure is, like at a very high level. And there are like tons of proteins that self-assemble, like 120 copies or 180 copies, proteins self-assemble to make this large giant structure. This is another like really big virus, but you'll find pattern here in all these proteins. They self-assemble. Last time we talked about quaternary structures, right? And they will come together because those proteins are sort of not in a happy state, not in a like stable state, and then they end up actually forming this beautiful structure, and which ultimately obviously provides a function to all these viruses. Or they will preserve their nucleic acid, their DNA inside these viruses, and that's the function of these proteins, right? And but also like understanding the that's the functional part of this proteins. But understanding them, it also humans are very inventive, right? Human learn to build aeroplanes by seeing how birds fly, right? And obviously humans didn't try to fly on birds, and what humans did was then they learn how birds fly and then make something like that. And now humans have been flying for 100 years, right? And you can you can think of a world without flying, right? Without having all these flights and all, right? And in a similar way, like understanding this virus itself, it gives us new insight to build tools, how this virus do function, right? Viruses are really good at entering into cells, for example, right? They can take over cells, and that's what, for example, coronavirus does, right? And they will interact with our cells, and they can bring nucleic acid into cells. And just by knowing those things, knowing those features, understanding these structures, understanding the protein structures, it sort of, again, it gives us like lots of tools, lots of insight to re-engineer things so that we can cure disease, and for instance. And so this virus, for example, a lot of these viruses actually, lentivirus, we talked about lenti and adeno-associated virus in our genetic engineering class too. And a lot of these viruses are now being used because they have an interior where there's an empty space. I mean, usually those are not empty space. Usually those spaces are filled with their own nucleic acid, own genes and things like that. But you can re-engineer these viruses to make those spaces empty, and so that you can load your own stuff or your genes that you are interested, right? And people have been re-engineering all these viruses to carry basically, for example, CRISPR, or gene editing, or for gene therapies and things like that. And this is really this is a kind of extraordinary. I mean, obviously, again, we take it for granted, and a lot of this knowledge and a lot of these technologies, we think, okay, it's great. But how humans over hundreds of years came to realize that this species, these life forms, can be used for something beneficial in our own life, right? And and then again, for that, it requires a lot of engineering, a lot of understanding, and protein engineering has been at the forefront of re-engineering all these viruses. Obviously, when you enter into the field, you'll learn more, and this is only very brief. And we all talked about coronavirus, right? And one of the features of coronavirus is that it has this spike protein, which looks like a monster. It's a huge protein, like for protein engineers us, it's so complicated, so much is here. And then in this spike protein, and as you know, that this is one of the major reason how coronavirus can enter into our host cells, and then take over the cells, right? And lots of people died. And and this virus is also really good at keep mutating itself. And not only this virus, most viruses, they mutate, so they have protein machinery that can make them mutate faster, and that's their survival strategy, right? If they can mutate faster, they survive better. If they're slow, dumb, and they cannot mutate, then go to extinction, right? They will die out. And so because of that, this is their survival strategy, to mutate so rapidly. And And because they can mutate, and most of those mutations again happen in this loop region, right? And the reason why it happens is that these regions are very flexible. And if you mutate in this region, what happens is that this protein can then switch their invasion pathways, for example, over time, or they can have a better affinity towards the host cell, right? And a lot of these mutations, because of all these mutations, these new strains keep coming. We have heard about like Omicron and Delta and all these different variants, and all this obviously again come because of the fact that they can mutate in important regions of the protein, and a lot of this. Because of these mutations, there has been constant need of making new vaccines, right? So that you can neutralize these mutating regions. Again, so this is for people who do protein engineering. This is a big both sort of challenge as well as opportunity out there to have a better protein engineering approach, so that you can build vaccines or other medicines, sort of things. Those are very few examples of very sophisticated level of protein structures that I talked about. And so now again, as I said, humans alone have how many? I said more than 25,000 proteins, right? And it's impossible to go and talk about everything. So if you have any favorite protein, any favorite machinery that you want to understand, you have the tools. All you have to do is find literature, and then I already showed how to use PyMol and then PDV data bank and find out those structures and just play with those. And obviously you'll amaze how beautiful and how interesting those protein structures are. So I would very briefly, I would talk about another class of proteins that people did not study for a long time. For a long time, scientists thought that in order for a protein to function, to have a function, it must have a structure, right? And that sort of makes sense. If you want to live in a house, it should better have a proper house structure, right? It cannot be a heap of bricks, right? How can you even live inside inside a heap of bricks, right? That's not possible. And so because of that, for long time, our understanding was that we need to have a proper structure, three-dimensional structure, in order to have these proteins working. But very recently, well, that recently means about a decade and a half, or even 2 decades now, scientists found that there has been increasing evidence of these proteins that do not have structures, and they do function as well, and they have huge functional relevance. They do so many different things inside our bodies, and those proteins are called intrinsically disordered protein. I think some of you are asking questions, and I'm not sure if I'll be able to actually so yeah, so you can ask later. And I think Ipsida and Hafsa asking questions. So yeah, hold on to your questions. Let's walk through all the presentation, and then we can discuss about those questions. So this the intrinsically disordered proteins, they do not have proper structure, yet they do profound functions inside our cells. And the reason why intrinsically disordered proteins do not have function, because last class we talked about this energy funnel, right, where protein are made, and then they have they go down the hill, and they will try to find the the lowest energy state, right? And that is the state where the protein would be stable, and it will have the lowest energy, right? And so that's the case for most the proteins that have structure. And then once it gets to this energy state, it cannot climb up. And in order to climb up, to get into different shape, is that you need more energy. And then where are you going to get that energy, right? You have to either provide from outside somehow, I don't know. But the intrinsically disordered proteins, they do not have a very well defined this deep well kind of stuff. They have multiple stages, and they are there are multiple stages. They are slightly different in energetics. and because of that, it is very easy for these proteins to keep flopping around and not have a very well-defined structure. And so this intrinsically disordered protein actually play a big role in functions, like in all different ways, like during cell division, the centriole, like in disease, for example, in Parkinson's, in Alzheimer, all this plaque formation happens, like different genes get mutated, and because of that they form this intrinsically disordered protein, and that leads to the formation of clustering of proteins and things like that. And so it does play a big role. And then there's a new field coming up, which is called phase separation in proteins. How like different organelles are formed, or different mechanisms during, for example, during stress and things like that. And there are all these proteins, intrinsically disordered proteins, play big role in terms of functions. Again, I'm not going to go into details, but if anybody is interested in structureless proteins and how they function, you can always read about intrinsically disordered proteins and find out more functions. I myself actually work a little bit, not exactly intrinsically disordered protein, but when I was working at the University of Washington, I found a protein that that can form like very sort of unique behavior inside the cells. They can form what is called like non-Newtonian fluid type behavior, which is never seen in protein machineries. But things like that can happen. A lot of these proteins functions are still not understood, and there's a lot of rooms to sort of study and then engineer these proteins. So that is just to give you a little flavor how these protein machines look, and again, the universe is so vast and there's so much you can learn about. But we will move to this new chapter, this designing protein, engineering protein, and what can you do? How can you like engineer protein from this first principle, right? Just understanding protein, and what can you do for curing diseases, right? Again, we cannot cover everything. I will just try to give some example in like vaccine development and things like that. So the protein engineering is, as I said, it's a big field now, and it emerged in last 30 years. This is about 30 years, not even 30 years, I think like 25 years. And before that, there was no such thing like protein engineering. And as the protein engineering emerged, and there are two branches of engineering protein, and one is you can take protein from nature and slightly tweak. We talked about antibodies, right? You don't have to build a new antibodies. You can just take an antibody that nature made and slightly tweak it so that you can repurpose the function and you can fight new disease, right? That's one way. And the other way of designing protein is totally built from scratch. And like you don't know any, that protein does not exist in nature, and you just want to make a, from basically it does not exist in nature. You just want to make from scratch, like from very basic things. That's incredibly hard. That's like, but we'll talk a little bit about. And then two of the pioneers that actually did all this work, majority of the work in this is David Baker, who works in the front of like building proteins from scratch, basically, like that protein that's never existed in nature before. And then the other kind of protein engineering that I talked about is to tweak the existing proteins and have new functions or have better functions. And those work mostly it started by Frances Arnold, who actually got the Nobel Prize in 2018 for that very influential work. And especially the Arnold's work, what we call directed evolution, meaning that you evolve the protein to re-engineer so that you can reshape and you can have new function. It's just, nature has been doing this kind of evolution, right, over billions of years to have new functions and things like that. But you can do in the laboratory, and that work is very profound, and most of protein engineering in the industry these days will use this kind of directed evolution, and obviously that's why Arnold got the Nobel Prize in 2018. And for proteins to make from de novo, it is extremely hard. De novo means like totally new. It never existed before. And obviously David Baker, he tried doing, 1998, he started new, programming method and things like that. And in over 20 years, he has been extremely successful. And David is at the University of Washington Institute for Protein Design, and Arnold, she's at Caltech. I actually work with David Baker. So I always give the analogy of, like how building protein, like building de novo protein is like building a brand new car, right? That car never existed before, and you just wanted to build a new car, like from scratch, right? Which is very hard, I can tell you. And then Frances Arnold's approach would be, like, let's say you have a car, old car in your garage, and you just want to like fix it somehow. And that again, like being able to repair an old car also has huge importance, because you can fight diseases in a new way. But this one, from challenge perspective, I would say, like, really, really challenging. And but again, these people started the work, and we have, and so the reason why de novo protein design is extremely hard is because we still do not have, well, now we have some better tools and understanding how protein folds, right? In the last class, I said, protein starts with this long chain of amino acid, and that it miraculously, it folds into a three-dimensional shape, right? And it folds because I said that all these atoms, all these side chains and molecules, they want to be happy, they want to go to the lowest energy, and because of that, they get to the lowest energy by folding, by forming a three-dimensional shape, right? So that everybody's happy there. And because there are hundreds of thousands of atoms associated with one particular chain, like one regular, like for example, insulin protein has about like 100 some amino acids, right? And each amino acid will have, I don't know, like 20 atoms maybe, or even more. And you can imagine like how many, how many thousands of atoms you will have per proteins, right? And each atom can interact with other atoms in a different ways, and they have all their own energetics and things like that. And because of that, it is extremely challenging to understand this protein folding process. And precisely, like if you are given a sequence of, let's say you are given a random sequence, and if you're asked to tell what would be the three-dimensional shape, the final three-dimensional shape, it was extremely challenging. 20 years when David Baker started this work by building new software called Rosetta, it was just beyond like human imagination what how you would be able to do that, how you would be able to predict that structure. But if you can do that, the good side is that then you can build new proteins, right? Because if you know a sequence of protein amino acid sequence that will predictively fall into three-dimensional shape, then you should be able to make any proteins, right? I mean, that's the logic, right? And obviously it took very long time to build something, and that building means you have to build softwares which can do the calculation so that you calculate the total energetics. And this sequence alone can have millions of different structures, right? Different folds, right? And like what is the lowest energy fold, which should be ultimately the structure, right? And just those kind of calculations was extremely hard to do. It required supercomputer and things like that. But David Baker, who made this tool called Rosetta, and in 20 years it was able to at least predict protein structure. And then last year there was big like it's sort of, people are saying that this has totally revolutionized. It is going to revolutionize, and no doubt about that. Is that two things came up. One is this David's effort to build Rosetta over last 20 years. He improved the Rosetta, and he, David and another colleague of mine at the University of Washington, actually, they built a new tool called Rosetta Fold, which can predict protein structure in a way better way. And then the DeepMind, which is a Google company, and DeepMind also came up with this AlphaFold 2, which again, took the entire scientific world by storm. and there's like so much talk about AlphaFold and how it is going to revolutionize the medicine. And I have to be very honest that it is going to revolutionize the medicine, because being able to predict protein structure and being able to make protein and engineer protein is such a demanding and such a huge thing in the field of medicines that you will be able to appreciate only if you enter into the field. It's very difficult to um, the importance of all this work. And so this Rosetta Fold was Ming Yang, actually, who was also my apartment mate, and she was my co-worker. And David Baker, they developed this Rosetta Fold, and then Demis Hassabis, many of you already know, he is very influential now. And John Jumper, they built this AlphaFold, and AlphaFold is like already now widely used in medicine, in companies, in universities, in everywhere, basically, just in 1 year. Sometimes technological revolution comes in like in a storm, and it just basically it takes over everything. And so the reason I wanted to mention about this Rosetta and Rosetta Fold is that now that if you can predict protein structure, you should also be able to build, right? And that was a point that I wanted to make. And the way we do is that we build these shapes, like the way carpenters will build like some kind of shape, like you want to build a house, you have to build the structures and things like that, right? First you just have some piece of wood, and then you basically put more things, like you cut, make holes, you point and things like that. And if you do all those things, you can bring like 2 pieces of wood to make, and then you can build something, right? You can build a house or any furniture, right? And so same thing happens for proteins. First thing you have to do is that you have to build the mold, right? You have to have the basic structure, like for example this alpha helices. So you can mold those, and then you have to make sure that this stays as it is. And what would you do if you're a carpenter? So you'll first build, let's say you are going to make a table or something like that. First you'll shape all these things, and then you'll stick something, you'll nail it, right? You'll put something so that it stays there. And so same thing happens for designing protein from scratch. So you build the basic folds, using again Rosetta, so that you can compute. And now obviously people, I'm not sure if that feature is available in AlphaFold yet, but at some point I'm hoping that people will use AlphaFold as well to build proteins. And then you can assign all these side chains, the amino acids that I talked about, all the sticky side chains that I talked about in the last class. And you can assign all those to give this rigidity, right? And then you'll have a very rigid structure, right? And then that will hold this protein structure, just the way that when you build a table, once you nail it, once you put all the required things, it'll just stay as a table, right? And so that is what. And so far Rosetta is the only suit that people use. And again, Rosetta was built by David Baker about 20 years ago, and it's a software package that does all these calculations so that it can build proteins. And so intuitively, I mean, it is simple, right? It's just, as I said, I keep talking about being a carpenter, making making new structures, right? The only problem here is that these are all molecules, right? And how do you fix things rigidly? How do we make how can you become a molecular carpenter, right? It's hard, and because of that you need all these tools, that different kind of tools, like computational tools, molecular tools, atomic tools. But if you have those tools, you can build. Why not, right? And I myself work in making using all these tools, and making large protein structures. For example, we talked about how virus is made, right? It is made from 120 proteins. How did that even happen? And so it turns out that nature also use all these different tools, and they call it evolution. Obviously evolution is a tool for them, and so they basically randomly keep sampling so that they can make bigger things. And so in a way, with all these tools that we have now, like Rosetta and other AlphaFold, hopefully, and you can bring things together, like just the way the carpenters work. You can also bring proteins together, like these are all alpha helices, and you can superimpose, and you can make like copy and paste, and sort of use like some tools to fix them, right? And if you I'm just showing here like how you can build a case like virus like case from simple proteins. It's again the analogy goes to the carpentry, and so this is my own work, and we try to build something like that is the shape of size of viruses. And then why would you like to build all these things? Because this have huge importance in like vaccine development and things like that. And so again, I'm not going into very details, but with this all these tools, with mathematical help and all, you can basically do this kind of stuff. And so I one of the work that I did was we built all different shapes, protein structures, which can be again further used to build larger things, and then you can have functions. And even right now I'm using a lot of these proteins to do like engineering stem cells and things like that. And these are some beautiful proteins that we made. I mean, obviously here it looks like some nice shape, but then as I said, in protein the shape gives the function, right? You can derive new function based on the shape and their functionalities, kind of stuff. And then we also build like really large things, like this looks like a crown, and this is like small case, which is a symmetry of tetrahedron, and it has like 24 chains of protein that self-assemble to form that quaternary structure. Again, I just want to tell you that these proteins are from total scratch. None of this protein existed in nature. We build it from total scratch. And evolution, it took like billions of years for them to make this. But as a modern scientist with all these tools, you can just build in a couple of years. And then this is an example of we call icosahedron case that we made, and this is the model, and this is the experimental, and again this is using like Rosetta tools, and build from total scratch. And again, I keep telling, I mean, at this point maybe you won't be able to find any functions, but then again, this like cases can be like used for vaccines and things like that, and which I'll come to it later. And then we also built tools that allowed one of my colleagues, Ravi Divine, he took the antibodies that we talked about, right? Antibodies can fight disease, and you can re-engineer antibodies, right? And so one thing happens in nature is that these antibodies fight by themselves. Like you can imagine this one single warrior, one single army trying to fight. But you can imagine, what if actually you can bring a lot of them together and form a like big cluster of antibodies, right? How would they fight the disease, right? And again, the nature did not get enough time to actually explore these possibilities, fighting in numbers. And from our basic intuition, it says that like if you fight in big number, there's a better chance that you will win, right? Rather than fighting alone. And so what Robbie did was that he took antibodies, and then using a lot of this, again, proteins that build from scratch, and he was able to build like large protein structures, like these are like again, it looks like a virus, but it has antibodies in it, and it has like very precise three-dimensional shape and structure, who's and then he used all of these antibody cases to like fight cancer better, fight like lots of infectious diseases better. And it just it is marvelous how all of these things can be done in laboratory, and nature has been trying to do it for millions of billions of years, but with all these new tools you can just do in laboratory. And yeah, and then Robbie published this work in Science. And so that's sort of uh, de novo. I just showed a little bit. Obviously there you can explore more by reading. You can there are like tools available. You can visit like Rosetta Commons website and try to learn more how people use Rosetta to build new protein and all these things. And so it is not possible to like include everything here. But I just like, now that we can build new proteins from scratch, now that we can design proteins, that did not exist in nature. Like what kind of function you can give to this protein, right? What can you do? What kind of diseases you can fight? Again, the limit is only the sky, what you can do. Like, for example, as I said, I'm not going to talk in this talk, but with all this design protein at Harvard, what I'm trying to do is like re-engineer like cells, like T cells from like skin cells, and convert them to like hematopoietic stem cell, and then T cells, or even like natural killer cells, and hematopoietic stem cell, which can be used for bone marrow transplantation and things like that. And a lot of these proteins that I designed, those you can use. You can assign function, you can have a function, and then use in different purpose, and all applies to medicine basically. And so today I'm just going to give one example how these engineered proteins that did not exist in nature have been now been trying to use in medicine basically. And so this is a protein case. Again, it just the inspiration was the virus, obviously, because viruses have all this icosahedron symmetry, and it has like like trimers and pentamers, and then they assemble together, right? That's how the viruses are built. And following this inspiration, in David Baker lab, back in 2016, the first protein cases were built from scratch. And so when this was built, and obviously this looks amazing, and again, doing from scratch is like, it's a very grand challenge, big problem and all, but this by now, all of these protein cases are actually in these are explored in medicine, and this is like more close-up view how complex these protein cases could be. And this is like the icosahedron case here. Sorry, this is the pentamer, and there are like trimers, and this icosahedron case has been actually used to design a vaccine even against coronavirus. And so this vaccine, when I was at IPD, I also briefly worked a little bit, not exactly for this one, but related work. And this, the power of this vaccine is that, as you can see here, that you can use all these proteins to basically, attach new proteins, and you can like attach all these for example, spike proteins into the surface of this nano case, right? And if you take those spike protein from coronavirus and put into these proteins, you can basically essentially make a vaccine, right? And so that's the vaccine here. And at the core, what you see is that this, you see these pentamers, right? And then there is like these trimers as well. So it's coming from this, but all this, the yellow and like brown and slightly red, these are all the spike proteins that you can attach to this scaffold. And then the Science magazine called this is a dream vaccine, is because the reason they said this is a dream vaccine, because you can build this vaccine, and you can have so many different variants of spike proteins put into here, and you can make a universal vaccine that can actually fight against many, the entire pan coronavirus, for example, right? Not necessarily against one strain. And most of the current vaccines that we have today is that they're good at neutralizing one strain, right? But then the new strain emerged because of mutations, that a lot of these vaccines fail, right? And so one strategy could be then use this, the core of this protein case, and then attach different strains of spike proteins, so that it can neutralize so many different coronavirus. And that vaccine is obviously still in the pipeline. It is not available in the market. Hopefully, I think it's in phase 2 right now. Again, this is developed by Institute for Protein Design at the University of Washington. But also, sometimes in science, things move so fast. And I said, like, this work was done in 2016. The paper came out, and from 2016 now, what, 2022, just in 5-6 years, we have this big vaccine candidate that is basically, if it comes out, it's ready to rock the world, right? And so a lot of these things, you are, again, I keep telling this because that's the purpose of this course, to encourage you. A lot of these things, when you learn and sort of you start with a new, some projects, and at some point you will see, like, you can do so much with all of these engineering projects, all these things. And I think that's the beauty of science. There's like this reward, and there is also, it is needed, right? It is needed for the humanity, right? So this is, again, this science published this paper, is calling this our vaccine as the dream vaccine, and then it's just showing how you can actually functionalize this core nano case with all these like spike proteins on the surface and build the basically better pan vaccines. And so this is just, I, because I keep telling this, I can speak about so many things, but it's impossible to speak about everything. And there has been different, not just like building vaccines, but like like immunomodulation, like fighting autoimmune disease. And there has been so many now design protein, and every day, from mostly coming out from Institute for Protein Design at the University of Washington. And it's amazing to see how technology is shifting into a new direction. And my guess is that in another, like 10-20 years, by the time you become mature scientists, there'll be a lot of things changing. And especially, like I talked about this AlphaFold, right? AlphaFold came out just last year, and in a year they have taken the entire scientific community by storm, and it's changing in so many ways. All these pharmaceutical companies, I mean, not only AlphaFold, even Rosetta Fold I'm talking about, like AlphaFold, they said, they announced that by next year they will have the entire known protein exists in this universe. They will have the structure, they will predict the structure, which is like 100 million protein structures. And that is amazing if we can do that, because like a lot of these diseases, we don't even know the protein structure, and we don't know how to design a new protein that can fight disease, right? And so all of these tools, it's a great time, great wonderful time to be in science, and you contribute, and obviously you have to build up your own platform, step by step. And otherwise, I feel like there's so much resource. I mean, 20 years ago, you could not have asked for this, right? 20 years ago, you could not have imagined that you can do all these things, right? Yeah. So I think it's as I said, there's so much thing to talk about, but it's impossible to cover everything. I think I'll stop here today, and then take questions. And today we have more time for questions. Yeah, and then I'll just stop sharing. Yeah. If Shida, and somebody already, yeah, a lot of people are raising hand, but there are already questions before in the chat box, and I think there Ipsita and Hafsa. Can you, both of you, ask your question unmute, or if you still have that question, and then I'll come to Akila.
Sir. Mm-hmm. Sir, Othani Huda question too, that was actually intended for after the class, okay, not to disturb you in between.
Yeah, that's fine.
Yeah, sir, I have two questions. Firstly, why is mutation in human genetics considered, or genes considered as a mistake, but mutation in viruses is considered a survival strategy?
Yeah, no, I mean, it is only our interpretation. You can say mistake because there is a genuine mistake in the machinery, and both in the virus, for example, they have a so like when this the gene copying happens, right, and there is a machinery that copies the genes, right, it makes the, and so all these are machines, right, just like any other modern machines, right, and those machines can make mistakes, and the virus machinery is prone to make more mistakes. And the benefit of that is that, I mean, you can imagine if the virus has a machinery that is like perfect, like there's no mistakes, and you don't allow mistakes to happen, what will happen? They will not survive, right? They will die basically, because they need variations, they need to evolve so that they survive. And for same way, for our human purpose, if you have a machinery that mutates so fast, and what will happen? We'll have all these diseases, right, and we'll die. So for us, we don't want a very, but again, like there are very mutation. This is somatic hypermutation that happens in B cells, where all these antibodies are made, and that is a very mutation prone process, because if you don't have that mutation prone process, then you cannot make a diverse set of antibodies, right? So it all depends on the fitness, like, ultimately, like whether it is serving the purpose or not. It could be either, like you can have more mutation or less mutation, but whether it's benefiting the species or not, right? So yeah.
So like it's like isn't it like for one species mutation is beneficial, but again for the another species mutation is not beneficial. Like how is nature choosing that?
It's so nature didn't do anything. All it happens is, like, if you have something good, you survive. If you don't have, then you die, right? And that's how evolution happened. There's like no here the reward is the survival, right? So if somebody, because of mutations, if they survive, then they will take that. Like it's just like in normal life as well. Like, some a lot of people steal things and they survive by that way, right? And if that is good for their survival, they will keep doing that, right? But if that leads them to the jail, or like even like not existing anymore, then that'll go away. So it's just like that. Yeah.
Okay, sir. Thank you. So my second question is, so when we're talking about entropy, in chemistry, high school chemistry, we learned that the more the higher the entropy is, the more the disordered,
Yeah.
the randomness, and the more stable the system is. But the way our life like progresses towards, like from mRNA, how the information is processed from mRNA to polypeptide and to again to uh, 2D 3D structure of protein. If you see that in that progression, there is reduction of entropy, which means the stability is decreasing. And
Yeah.
so how
So
how
it
is it
all, yeah, I think I got your question. So it all depends on the circumstance, right? I mean, sometimes by it's just like at your house, right? Either you can, like, all the member can go crazy and keep dancing, or you can stay quiet, right? And if you keep dancing, it's obviously that's a high entropy state, and you might think that is good. I mean, it's circumstantial, right? At some point you have to be like, sit quietly so that everything happens nicely. And a lot of
Mhm.
these molecules actually, all these, like, atoms and molecules, they always want to go to a stable state, so that if they do not go to the stable state, what happens is that they are not happy. They want to find a stable state, right? And they will try to find partners, and so that they can interact, and that's how all these interactions happen, right? And the whole point of interaction is that so that they can save energy and go to the lowest energy. I think that's the that's another principle of nature. Yeah, this is all
So,
conflicting, obviously. Yeah, but
so you
that's how
mean
it
to
is.
say that for death, life is necessary?
Yeah, yeah, yeah. So,
Okay, thank you.
right. Rajushi, you raised before. I think you had questions, and then I can come back to Akhil after
Yes, I have a question about antibody.
that. Mhm.
That is, some antibodies are used as drugs, such as for COVID diseases. Some antibodies was tried as drugs. So my question is, if the antibody is injected as drug, won't the our body create some antibody against those antibody proteins? And when the second dose of the drug is injected, those antibodies will neutralize the drug, and it will deactivate the drug. What strategies are taken so that is there any structural modification in those drugs, so that once they are injected, our body will not produce
Yeah,
another
yeah,
antibody against them?
I got your point. I think you are asking about immunogenicity, so that we put into one single word. It's called immunogenicity. What is the immunogenicity? Immunogen, okay, whatever. Um, Basically, whether it's immunogenic or not, right? And so when a foreign thing comes to our body, then our immune system reacts, and it will create a memory, and then it will fight back, right? And so that's an excellent question. And a lot of these antibodies are actually I can actually, like, just give me a second. Let me stop